Assalamu alaikum students. In this video we will discuss about the de novo fatty acid synthesis. We will cover this topic under the heading of the site, transport of acetyl CoA, requirements, activation, pathway and its regulation. So let's start. Students, the site for the fatty acid synthesis are liver, intestine, adrenal cortex, adipose tissues, and lactating mammary glands. It is a very important topic. The main site for the synthesis of fatty acid are liver, intestine, adrenal cortex, adipose tissues, lactating mammary glands. Now transport of acetyl CoA. Acetyl CoA, it is present in mitochondria. The two main sources of acetyl CoA are pyruvate. Through pyruvate dehydratase complex, acetyl CoA is obtained. This process occurs in mitochondria. And beta oxidation of fatty acid. These two are the sources of acetyl CoA. Now acetyl CoA, it is present in mitochondria, and the organelle required for the fatty acid synthesis is cytosol. So transport is very important. Acetyl CoA, it gets attached with oxaloacetic acid and forms citrate. Because acetyl CoA it can't cross mitochondrial membrane, so that's why acetyl CoA gets attached with oxaloacetate. Now, mitochondrial membrane is permeable for citrate, and citrate with the help of ATP citrate lyase, citrate crosses the mitochondrial membrane, and in cytosol with the help of ATP citrate lyase it forms acetyl CoA and oxaloacetic acid. Now acetyl CoA is in cytosol. Students, I am repeating again. The two main sources of acetyl CoA are pyruvate and beta oxidation of fatty acid. In mitochondria, this acetyl CoA is present. The main site for fatty acid synthesis is cytosol. And acetyl CoA gets attached with oxaloacetic acid and forms citrate. Citrate can easily cross mitochondrial membrane, and in cytosol, with the help of this enzyme, it forms acetyl CoA and oxaloacetic acid. In this way, the transport of acetyl CoA takes place. Now, the requirements for acetyl CoA are is acetyl CoA, NADPH for reducing equivalent, and ATP. The requirements for the fatty synthesis are acetyl CoA, NADPH, and ATP. Now coming to the activation of acetyl CoA. Activation. Activation. Acetyl CoA with the help of acetyl CoA carboxylase enzyme, it requires ATP, biotin, and carbon dioxide. It converts into malonyl CoA. In this way, activation of acetyl-CoA takes place. Acetyl-CoA, with the help of acetyl-CoA carboxylase, converts to malonyl-CoA, and this is a very important regulatory enzyme. And this is a rate limited and committed step. Now coming to the pathway. Students, pathway, there is a very important mnemonic to understand the pathway of fatty acid, and you can easily understand and write in your copy. ATM transaction kit started refund of delivered and cash rupees through ACP. Here AT means acetyl transacylase. MT mean malonyl transacylase. KS mean ketoacyl synthase. R means ketoacyl reductase. D mean dehydratase. ER means enoyl reductase. T mean thioesterase, and this is the acyl carrier protein. This is a very important mnemonic. ATM transaction kit started refund of delivered and cash rupees through ACP. Acetyl transacylase, malonyl transacylase, ketoacyl synthase, ketoacyl reductase, dehydratase, enoyl reductase, thioesterase, and ACP. Now coming to the pathway. Acetyl CoA, it gets condensed with enzyme cysteine sulfahydryl group. With the help of enzyme acetyl transacylase, and there is a transfer of acetyl group to the cysteine of this enzyme and forms acetyl enzyme and COSH it released. This is the first step. There is a transfer of acetyl group to the cysteine of enzyme, and there is a formation of acetyl enzyme and release of COSH with the help of enzyme acetyl transacylase. In next reaction is malonyl CoA. It gets condensed with ACPCSH, acyl carrier protein contains sulfide group, and it leads to the, with the help of enzyme malonyl transacylase. This is the second step. There is a transfer of this malonyl group to the ACP, and it leads to the formation of malonyl ACP, and there is a release of CoA acid. This is the second step. In third step, acetyl enzyme and malonyl ACP they gets condensed. In third step, acetyl enzyme and malonyl ACP they gets condensed, and it leads to the formation of beta ketoacyl ACP. And the enzyme that is required is ketoacyl synthase. There is a release of CoA and carbon dioxide. Is the third step. I am repeating again. First step is acetyl CoA gets condensed with enzyme cysteine sulfhydryl group with the help of acetyl transacylase. And in this step, there is a transport of acetyl group to the cysteine of enzyme, and there is formation of acetyl enzyme and release of COSH. In next step, malonyl CoA gets condensed with ACPSH, and with the help of enzyme malonyl transacetylase, there is a transfer of malonyl group to the ACP, and there is a formation of malonyl ACP and COSH. In third step, in third step, there is a condensation of these two acetyl enzyme and malonyl ACP with the help of ketoacyl synthase. There is a release of CoA and carbon dioxide. There is a formation of beta ketoacyl ACP. Now, in next step, with the help of ketoacyl reductase, it requires NADP. NADP plus converts into NADPH NH plus, and there is a formation of beta hydroxy acyl ACP. Okay, students. In next reaction, with the help of dehydratase enzyme, there is a release of water molecule. Dehydratase enzyme. This beta hydroxy acyl ACP converts into enoyl ACP. In next reaction, this enoyl ACP, with the help of enzyme enoyl reductase, there is also utilization of NADP plus, NADP plus, and converts into NADPH and H plus, and it converts into enoyl ACP. Enoyl ACP, it converts into the butyryl ACP. Okay, students. Third step. This one is fourth step. This one is fifth step, and this one is the sixth step. Students, third, fourth, fifth, sixth step, they are repeated again and again. So there are repetition of 6 cycles, and at the end, this butyryl ACP converts into the palmitate, which is a fatty acid, 16 carbon atom containing fatty acid. It means there are repetition of total 7 cycles. In this way, fatty acid synthesis takes place. Students, beta ketoacyl ACP, with the help of ketoacyl reductase enzyme, it converts into beta hydroxyacyl ACP. It requires NADP plus, which is a reducing equivalent, converts into NADPH and H plus. Beta hydroxyacyl ACP, with the help of dehydratase enzyme, it converts into enoyl ACP. Enoyl ACP, with the help of enoyl reductase enzyme, it converts into butyryl ACP. And third, fourth, fifth, sixth step, they are repeated again and again. It means there are repetition of 6 cycles, and ultimately, the next step, it is converted into palmitic acid or palmitate, which is a 16 carbon atom containing fatty acid. And in this way, there are total 7 cycles. And there is a mnemonic to understand the pathway of the fatty acid synthesis. Students, now coming to the regulation of fatty acid. Regulation. Acid regulation takes place by covalent modification through phosphorylation and dephosphorylation, and also through hormone. Acetyl CoA carboxylase enzyme This enzyme activates only in the dephosphorylated condition, and insulin and thyroxine, both hormone, they favor this condition, diphosphorylated condition. In this way, acetyl carboxylase activates. While glucagon hormone, it favors the phosphorylated condition, and it favors the phosphorylated condition, and it inhibits the acetyl CoA carboxylase enzyme. And in this way, the fatty acid synthesis are regulated. Through gene expression regulation, the fatty acid synthesis is also regulated. Okay, students, this was all about the fatty acid synthesis. Keep watching and thank you.